year of, I guess, traveling to Wrigley, right? Or like a few months, every boat ride back and forth.
Yeah.
Levi is the only person I know who tried to fight seasickness by reading books down in the bottom, in the bottom of the, in the belly of the Miss Christie. That was very heroic.
Because they wouldn't let me out on the back deck on Thursdays because Thursday's a cargo day.
Right, yeah. So, and Levi published some great papers and found postdoctoral position, and hopefully will be staying in contact with us doing environmental research and teaching, which is his passion. And Levi, with that, I will transfer powers to you.
Yes, power. All right, cool. So I am going to share screen. Okay. All right. Welcome, everybody. So I just actually want to open up with an inspirational quote that's on a mural in that I saw many times running. And it's actually on Fountain in Los Angeles. So I actually really like this quote because I think that it speaks to my need that the answer to most questions in the universe are usually given with a deadpan simplicity when all said and done. And I think Betty Davis was right about a lot of things, but also this. And anyway, actually I was running on Fountain this morning as well. So, okay, so I'd like to read from a series of prepared statements. So this is my thesis talk, my defense, new measures of biological diversity for stream monitoring. But before I get into like all the details of it, I want to just preemptively thank a whole bunch of people. So I want to first thank my parents who basically let me do, like actually pursue this kind of stuff as my career and like kind of just let me do what I wanted. And we're really good at instilling a work ethic in me, which some of you probably know about. And I want to thank Stacy. Stacy's right here. So we've been together for over 13 years now, and this is the kind of stuff we do. And she's been really great and supportive. I want to thank Team Newsgen, which is the best lab ever, including 316B, which is now 316D, which is now Gary Anupam and I in our living rooms. I want to thank my committee for guidance and a lot of the help I've actually received from also people in your labs individually. Folks over at Squirp, and I will explain who they are in a little bit. I also really want to thank CrashSpace, who take up a number of photos here on my my friendship collage. But CrashSpace is this great hackerspace I've been part of for over 10 years now in West LA. And anyway, it's been really influential in my life. I also want to thank a lot of my students, a number of whom are on here right now, some of whom are in grad school themselves, which makes me feel kind of old because I remember teaching them when they were like 14, but whatever, so it goes. And I also want to thank the EPA, which is continue to exist despite a whole number of things. Anyway, so without further ado then, let's go back to me. Uh-oh. And all right. So new measures of biological diversity for stream monitoring. So like this is 3 years of my life's work. And here's kind of like an overview of it. By the way, the cartoon over here kind of speaks to my background in physics, where you spend years and years of your life and it boils down to something you write on an index card and nobody cares. All right. So why did I do this? I want to get into that. The first chapter, so the first paper here, is this concept called zeta diversity. And those of you who work with me in the Newsman lab are really tired probably of hearing about it. Too bad. The second one is actually this chapter was originally what inspired me to go down this route. And I'll get into like why I'm interested in networks. But the one time about the topology of co-occurrence networks and how they relate to these ideas in ecology. And then I want to come back to zeta diversity, and this time using some really fancy genetic sequencing and environmental DNA work. And then after all said and done, like, why did I do this? All right, so I'm gonna open off with what I hope is kind of obvious, is that streams are important. So they're really important. There's an interface between water and land carrying nutrients. supporting everything in terms of like human systems from agriculture, just mediating environmental quality. And then biogeochemical cycle is just like one of those catch-all terms that I really love to use. But human beings absolutely depend on it. I mean, like civilization grew up around rivers, but we also have a lot of negative impact on river ecosystems. So we haven't necessarily been that great stewards of them throughout human history. And actually one of my former students who's on right now has also worked a lot on streams, can probably also speak to that. But this is about me. So, so who will speak for the streams? In California, at least, we have the Southern California Coastal Water Research Project, SQUIRP. And SQUIRP has been around for over 50 years now, and they do a lot of monitoring of the health of streams in California. And this also includes coastal California, so looking at marine sediments. We also had the opportunity to look at some of their work there as well. And they do all this stuff. They oversee all this work, environmental monitoring. They've been really great to work with. I've learned a lot about this, considering, again, as Sergey alluded to, my background has nothing to do with ecology. And I've learned a lot from working with them on, like, what is this stuff? Why would anyone care? Like, what's the nitty gritty of actually carrying out this work? Yeah, all right. So specifically, one of the things that SQUIRP does is they look at the health of streams. And the index that is used is called the California Streams Condition Index. And this flow chart is sort of like, I'm trying to do the best graphical representation of how this works, but you take samples of river bottom muck, you sort through, you look at all the invertebrates that live in there. And then you just put things in like different piles of like, okay, this genus, this genus, this genus. And then you look at the patterns of, you look at the distribution of diversity. So like how many of each type of genera do we have? Compare it to places that are relatively undisturbed. So these are called the reference sites. And then we look at functional diversity, and I'll talk about what functional diversity is in a little bit. And we look at taxonomic diversity, which is just like literally how many different genera do we have. And then a bunch of math, and then we get this index that comes out of it. And this is a technique we use to say like, okay, how degraded does a stream look like? And this sort of method actually, so this is the California Streams Condition Index, but this is the kind of method that's actually used in doing all sorts of indices of the health of streams in various places all over the world. But as you can kind of imagine, this first step is a rate limiting step. This takes a lot of work. So as you can imagine, going through, looking, getting all these invertebrates and sorting them down to genus takes a lot of time and effort, and people that are really, really trained. And I say this having done work where I've sorted insects down to order for a different project, and that was relatively tedious for me. And I don't really have training in it, but I imagine even with training, this is a huge amount of time. And it turns out, like on average, it's about 9 months to go from start to finish with a particular set of samples. So this is where we have the motivation. There's got to be a better way with this. And luckily, we now have the technology to find a better way. And it turns out we can try to avoid this step. So no more just sitting there trying to count these things by hand necessarily. We can go and approach this better living through metabarcoding technique. So how does this work? So instead of trying to sort everything by hand and go like, okay, this looks like this genus, this looks like this genus, and so on, we can take an environmental sample. So let's say like river bottom sediment, extract the DNA, and there's lots of kits, by the way, for those of you who haven't worked on doing library prep, I highly recommend it. But it's to extract the DNA, and then what you do is you clip out certain regions, and then you amplify them, which is basically you make tons and tons of copies of these. You sequence it, and then once you sequence it, you look for these, like you look at these little snippets and go like, oh, this snippet looks like it belongs to this organism. This snippet looks like it belongs to this organism. And then you can much more quickly take and process all these samples and then organize and get your community profile. So this is, instead of having to sort everything by hand, you throw everything in a blender, extract the DNA, sequence it, and go like, okay, this, this, and this. So this has gotten appealing because we've gotten a lot more efficient at doing sequencing, and the computational technology has gotten a lot more efficient in recent decades to do this in a practical sense. So this is where we were back in 2017. This is a joint proposal with SQUIRP and USC to essentially develop like, okay, how do we make an index then that can incorporate this meta barcoding data? So we have our index that we built around hand sorting everything. Can we make assessments of the health of streams that can incorporate this? Because if we can do that, then we can take something that instead of doing 9 months, we can do something in a few weeks and much more rapidly assess stuff. So we can do stuff at a greater scale. And by the way, because you can also extract stuff with DNA, you're not limited just to invertebrates. We can also start looking at all sorts of other communities as well of biological interest. So we worked on this. This is 2017, and as Sergey also mentioned about going back and forth on the boat, I have a very distinct memory of my part of the proposal having to get it in under deadline and writing on my laptop on the boat trying to do this, getting violently ill, relieving myself at the back of the boat, going back down to the hold, finishing writing my part so we could submit it by 3 p.m. that day. So long story short, we got the grant, but then here's the problem is that when you get the money, you actually have to kind of do what you said you were going to do. So now you have the congratulations, now you have the money, now you actually have to do something with it. And so this was sort of how I felt. I found out when I was working on the island back in the summer of 2017. And it was really exciting. And all of a sudden I went like, what did I agree to? Anyway, let me backtrack a bit. So the timeline here is not totally linear. It's kind of like Pulp Fiction. But if we go back to 2003, and hopefully some of my REU students I've mentored who are in REUs are on right now. This is a really great life story, or at least I like to think it is. In 2003, I had a physics REU because I was an undergrad physics major, and I did it at Notre Dame in Indiana. And the project I worked on was a cosmic ray detector, and it was built specifically to look at the gamma ray flares from this black hole, this active black hole that formed in it's known as Cygnus X1. So the problem was the Cygnus X1 was an active flare in the late 80s. The array was built, and as soon as the array was built, Cygnus X1 has never flared since. Still to this day, it has not flared. So literally my entire REU was looking at something where nothing was going on. This was not a very motivating experience, although I did learn some more coding in Fortran, I guess. But more importantly, is across the hall, there was a lab group led by a mathematician or mathematical physicist known as Laszlo Barabasi, who did a lot of the early work, now this is getting on to 20 years ago, to start mapping the structure of the internet, which, by the way, this is the thing here on the left. The photo on the right, by the way, is taken from the Notre Dame campus. So it was mapping the structure of the internet, and I got really fascinated with that lab group, and I got really jealous, like, why didn't I intern with this lab? Because they were doing all this stuff they're talking about, like networks and structure, and there's all these things that are really cool to come out of mapping networks that seem to, like, you have these patterns that pop up over and over again, like whether it's social networks or biological networks. And this sort of like spoke to at least my bias, is that the universe runs a lot like the Taco Bell menu. There's 6 ingredients and they're permuted endlessly. And so there's a lot of repetition in the universe. So I thought they were really cool. I also learned a lot about another Hungarian mathematician, Paul Erdős, who is probably the most prolific mathematician in the 20th century. And you can actually find out how closely you are in terms of degrees of separation you are from him. Turns out I'm about 5 degrees of separation from Erdős. Sergei Nushin is 4. Okay, so back to present day. So we got this. Now they were like, okay, you have the money. Now you have to go do something with it. And my thought was, all right, well, if we're going to get DNA data, and that DNA can be invertebrates, but it could also be bacteria, it could be algae, it could be whatever. Are there ways that we can start looking at real general purpose tools? So like the Swiss Army knife of biological indices, where I can take communities, any sort of biological community, and start looking at patterns and say, like, are these patterns representative of healthy or unhealthy ecosystems? And I will have to admit, I chose this route because I'm a bad biologist. I just wanted to look at patterns because I'm at 30,000 feet with not a solid bio background. And I want to say, look, are there just things that look degraded from, like, basically from orbit? And it's sort of independent of whether or not we're looking at bacteria, if we're looking at bugs. So I wanted to set about to develop a more universal framework to do this. Again, this is like my bias. So this leads me to chapter 1, and this idea of called zeta diversity. So this is for when alpha and beta don't do, and I still have trouble writing that zeta character. Okay, so this is like first method like I worked on to try to tackle this question. And so coming back here to this idea of like, how is that California Streams Condition Index calculated? It's a composite measure where we look at what's known as the taxonomic diversity. So that taxonomic completeness, like, do we have just the genera of our invertebrates? How many of them that we would expect to see if the stream weren't degraded? How many of them are there? And then if we know the genera, we also have this, what's known as functional diversity. So these are, well, I should probably just explain what this is right now. So taxonomic diversity is just like, it's literally just like the number of unique categories. Functional diversity, and this is a quote, this is from, anyway, Joe Cocker, this is actually a great song. But functional diversity is really the number of unique ecological roles. So, and that can be, there's lots of different ways to classify it. In this case, it's what eats what. So it's like functional in terms of like, are these things piercer predators? Are they herbivores? Are they bottom feeders? Are they omnivores? There's all sorts of like categories of this, but that's the functional diversity. So it's sort of like your job, and then the function and the taxonomic diversity is your name tag. So name, where you are in the org chart. And so we look at these two measures to start talking about the health of ecosystems. So that's for an individual sample. Now, again, going back here to this idea of like just sort of these big patterns, 30,000 feet up in the air, I want to look at not just like what's there, what isn't, but also just like what it's called like a landscape diversity. So that is like, how are things similar or different between each other? And so the established method in ecology is something called beta diversity, where you're just like, you're comparing two sites at a time. So alpha diversity is like local diversity, that's the individual community. Beta diversity allows you to then look at between any two communities at a time. And it allows us to look at things that are gonna, what's influencing these differences, whether or not it's just geographic distance, why things are different, is it environmental differences? So there's all sorts of questions that people can investigate looking at this idea of beta diversity. So just comparisons, like how much overlap is there between any two places? So going back through this, so like with alpha diversity, with individual sites, we can use it to look at inferring the effects of just stressors. So these can be things like just disturbance from if there are forest fires, as it changes the landscape around the river. Human impact in terms of like we put impervious surfaces around the river, so that's going to influence how water drains into the system. And so we can look at disturbance at an individual site. Beta diversity, again, it's landscape diversity. So we can look at the relative importance of what can influence these differences, whether it's patterns of migration, dispersal, if it's just or if you have like big environmental differences between sites and that's why things look different. Anyway, however, there are limitations. Alpha diversity, again, it's a single point measurement, and we're comparing it against a reference. You kind of get in these like circular arguments with like, well, how do we know what a reference is? Well, we assume it's something that's not degraded. Well, how do you know it's not degraded? And at a certain point, you just have to say, look, it's not near human beings at all. It's probably undisturbed. And that's going to be our reference. And it's the best we can do, and it's a good way to sort of bootstrap this. But you were left with these questions of like, since we're always doing these individual point measurements, is how representative of this is of your particular area, all right? Beta diversity, you're limited at doing pairwise comparisons, just two sites at a time. And it turns out, if you think about, if you want to try to extrapolate beta diversity to lots of communities at once, like let's say 100, and you're doing pairwise comparisons, imagine if you have 100 communities and this one freakish beetle shows up in one sample. If you're comparing now these 99 other samples to that one, everything gets skewed because now it's like everything's different compared to this one site because this one beetle happened to show up and throw everything off. And so there's biases that come up in beta diversity because of that. And this was something that I was dealing with and trying to figure out how to structure, like, how do I want to like look at these big patterns, these ecological patterns across a landscape, because they have these certain limitations. And I found probably one of the more important papers, I guess, of my career up to this point which is zeta diversity. And I'm very proud of this figure, which is why I put it in the Wikipedia page that I also wrote about it. But this is from Quia-McGough 2014, and it's this concept known as zeta diversity. And zeta diversity is just a way of saying, like, all right, we can just throw any specifics about alpha and beta out the window. We're just going to make a real general case about looking at landscape diversity. So zeta 1 is, imagine again, if you have 100 samples, 100 places where you look at all the local biodiversity, zeta 1 is the average diversity in your 100 samples. So zeta 1, if you have 100 samples, will tell you on average how many unique species or genera or families are going to be found in any given sample. Zeta 2 looks somewhat like beta diversity, and it basically says, now take any 2 samples at random, what's your average overlap between them in terms of like, how many species do they share in between them? So what fraction of species are going to be shared between them? So again, if you have 100, you do all 2 pairwise combinations and you say like, okay, on average, what's my pairwise similarity? Zeta 3 is what's your average overlap between any 3? Anyway, you can kind of see where you can go with this. You can sort of go to arbitrarily large numbers of zeta. You're limited just by like your data set. And this is turns out this, even though this looks like something really basic and just Venn diagrams and like, who cares? We all dealt with Venn diagrams in middle school math class. It turns out there's a lot of really cool things that come out of this. First off is, before I get into like all the cool things you can do with it, is I really need to make sure that this connects back to previous methods. So whenever you come up with some new fancy tools in research or is you want to make sure that it's not going to give you some like weird discontinuity from like what came before. So it is completely backwards compatible. Alpha diversity and zeta 1 are related in that zeta 1 is just average alpha diversity for your samples. Zeta 2 and beta diversity are related by this formula. And there's nothing actually further than that, because it turns out you can't do any direct comparison of 3 or more samples using just alpha and beta diversity. But for the stuff that's established, alpha and beta diversity, it is completely backwards compatible. So just using zeta 1 and zeta 2, you can go back to what we used to do. But you can do a lot more now. And the reason, one of the big reasons they want to look at this is because when you start going up to these higher orders of zeta diversity, you can start looking at things that change across lots of communities at once. And why you might care about that is, again, going back to our like one freak beetle, is that if it shows up or it doesn't show up and it skews 100 results at once, well, now let's say if I wanted to look at what's the average overlap between 10 samples at once. Now, if you have that, then you're starting to average out like any big effects. So if you're looking at things that are what's in common between any 10 samples, then you're looking at things that really have to change your what's ecological base load, essentially. So if something is gained or lost on average between any 10 samples, that means it's got to be a relatively common species, because it's got to show up in at least 10 of those. And so you can start looking at things that are less biased towards these rare taxa. So now you can look at things that are really common, so your cockroaches, so to speak, which are ecologically important. So there's a lot of things that you can play around with in zeta diversity and look at how it changes. So as you can imagine, as you go up with zeta diversity, in terms of like what's the overlap, like how many species are shared in common between sites or samples you're taking. This can vary with just the order. So that, again, the order is like zeta 1, zeta 2, zeta 3. As you go higher up, in terms of like how many samples are you comparing at any given time, you're going to find less species on average are going to be shared, let's say, between 10 or 20 sites at a time than versus like 1 or 2. So there's going to be, you have relatively fewer number of species that are going to show up that are going to be held in common between most of your samples. But again, it's thinking about your cockroaches. There's not that many necessarily in your across your landscape. They may show up in everything. And so they'll show up in your zeta 10 and zeta 20, but in terms of like your overall number of species that you're looking at, there's only a few that are really doing that. Other things that influences, just geographic distance, which is not anything unusual, like looking at landscape ecology. As things get further apart, they tend to get less similar. You see the same patterns with different orders of zeta diversity. And we also can use this to look at differences in environmental conditions. So like, let's say, of all the rivers I have different average water temperatures, I have different levels of dissolved oxygen. that's going to influence what species are held in common, because ecologically, these are different environments. And so this is all done in using R. This is all, like this analysis is all done using the ZetaDiv package. It's a lot of fun to play with. And I'd like to give a shout out to Anupam for helping me figure out how to install in the cluster. All right. Okay. So one of the questions I want to address is this idea of community assembly, which we can do using Zeta diversity framework. And so what do we mean by community assembly models? So think about when you see a bunch of species somewhere in an ecological community, is how did they all get there? How does it all work? Is it just like, is it essentially the castaway model? Or is there some sort of pattern where like this species shows up because it's got a relationship to this species, so it's going to influence, if I see this species, is it going to determine whether or not this other species is going to take hold, or is it going to tend to exclude it? So things that tend to have like really similar ecological niches, either they competitively exclude, or like one can reject the other, or they may actually just differentiate by, this is niche differentiation, where two species do a divide and conquer, where like, okay, this one pursues this ecological niche, this one pursues the other. So I didn't use finches because that's an obvious example. So we're looking at warblers. But the idea here is that if we look at just within ecological niche here on trees, we have like just different habitat types within a tree. 3 different birds can essentially partition out and avoid competitive exclusion. And so niche differentiation is a model where what species show up tend to influence each other, usually by some functional relationship. We're like, okay, this is my role in the ecosystem. You can't claim this turf. So if you show up in this ecosystem, you want to survive, you got to claim a different niche. Whereas the castaway model is like, whatever showed up is whatever shows up. And we can distinguish these two using ecological data by looking at how does zeta diversity decay with order. So again, if we were to plot zeta 1, zeta 2, zeta 3, and so on, against just sort of like how many sites are we comparing at once, if it tends to decay exponentially, it increases the likelihood that we're just looking at a stochastic castaway model of community assembly, just like whatever shows up there. If it tends to decay via power law, which is just a different mathematical function, it's evidence of niche differentiation. And this is a really powerful technique because this idea of community assembly is really important in looking at a lot of ecological relationships. And zeta diversity allows us to do this. Okay, so I was building a lot of mathematical tools with this, and now I had a hammer, and I was in want of a nail, and luckily, Squirp had a bunch of nails. So my advice, by the way, for those of you who are partway through grad school, is use other people's data. It takes a lot of work to collect your own data, believe me. So luckily, Squirp, because they've been around for a long time, has decades and decades worth of stream data. So in this case, I got my hands on 23 years worth of data. This is California. This is thousands of samples covering the state. There is some repeats. So we have, about 4,400 samples. 2,500 of them are geographically unique. So there is going to be some repeat measurements. And it's scattered across 56 watersheds in the state. And the data is really rich. So I have 334 unique genera of invertebrates that are found in the streams. They're sorted to 8 functional categories of what eats what. And I also had data on what was our environmental stressor, so what was going to cause degradation in our environments. In this case, it's the amount of land that's dedicated to human activity within a 5-kilometer buffer, so upstream of our site. So picture where you happen to be taking the data on the stream, all the land that flows in that river within 5 kilometers, how much of that is dedicated to human activity? And we're using that as our measure of environmental stress. Because it turns out like how much is dedicated to human activity is sort of a great catch-all proxy. So whether it's agricultural land or just asphalt or whatever, it's all a good proxy for, are humans having an impact or are they not? Okay. So how do I process all this data? So this is where we get into fun with permutation. And what I did is at each of those watersheds, 56 watersheds across the state, I would randomly take 25 samples. I'd calculate a couple different orders of zeta diversity. In this case, zeta 1, zeta 2, and zeta 10. And I chose these orders, zeta 1 just because it's alpha, essentially it's related to alpha diversity, which is related to how we calculate the stream index in the first place. Then we looked at zeta 2 and zeta 10. Zeta 2 is going to give us measures of landscape diversity are biased more towards whether or not we see rare species in any given set of samples. Zeta 10 is now we're looking at more of the just your really common species that tend to show up. And I chose Zeta 10 because when I was playing around with like looking at how things decayed, Zeta, by the time you get to about 10, you're starting to get to this asymptote, like with how Zeta diversity decays with order. Once you get to about 10, it pretty much flattens out. So if I went up like Zeta 20, I wasn't going to get much difference in terms of information. And so this gives me 3 different parameters to build a model. And the reason why it's 3 is because I had Zeta 1, Zeta 2, and Zeta 10 for taxonomic diversity. So how many species, or in this case, genera, are held in common. And then 3 more for how many functional feeding groups or functional diversity are held in common. So I wanted to look at these patterns of diversity for both taxonomic and functional categories. And then I calculated the average amount of land use for my 25 streams that are like within a 5-kilometer buffer. So this is my average measure of stress in a watershed. And then I randomly permuted this. I did this 100 times. So I ended up with 5,600 sets of these calculations. So 5,600 sets of Zeta 1, Zeta 2, Zeta 10 for functional and taxonomic diversity. All right. And here's one of the results. Wow, what does this mean? Well, first off, graphic design is my passion. So this is the Viridis color palette. But this result here, if we look at the color coding is, we look at local taxonomic richness. And so we're looking at just local diversity versus land cover, so which is not a surprising result. And that is essentially, so we have high local diversity here, so high alpha diversity, where you have very little human impact, so percent of the land that's dedicated to human activity. And as you go further and further out, if it's almost 100%, you have much lower taxonomic richness, which isn't surprising. You would tend to expect less diversity in areas that are more impacted. But what became interesting to me is just the shape of this distribution, which is it falls off, and it falls off relatively sharply. And Zeta 10, and this is for taxonomic diversity in this case, this shows you that under stress, every one of these watersheds, every one of these samples where we got data for the community, they started to look more and more different. So local diversity collapsed everywhere, but it didn't converge. Everything failed in its own unique way, which I guess I'd later refer to as the Ana Karenina principle. But it was this thing that we're, after all said and done, we could say at the watershed scale, local diversity falls, but everything fails uniquely. And so it looks more different. And so I was trying to think of like why this was. So I went back to Venn diagrams because it's easy to draw circles in paintbrush. And this is my thought of like what might be going on here. And so this is just this idea of like local diversity. So alpha diversity for any given sample, alpha 1, 2, and 3, really simple case. I have 3 samples, I'm looking at their local diversity. And under stress, maybe what's happening is that, yeah, your richness decreases. So the size of the bubble essentially shrinks. But then also everything pulls back from each other. So everything looks more unique because there's just less overlap between anything else. So it's degraded and you have this like greater sort of overall diversity, but it's sort of, it doesn't mean things are going great. It just means everything sort of failed kind of randomly. And in doing a lot of work on this, I found that there was an example. So people saw this a few years ago. This is this paper from Hawkins. They saw something similar, and they were just looking at it with alpha and beta diversity, but they did see something similar to this with patterns in invertebrates and streams that were under stress. And which was somewhat of a sanity check for me. It's like, okay, like I want to, like, at least somebody's seen this. So, and this is sort of like going at now talking about this idea of community assembly. When I looked at how does data diversity decay with order, so coming back to that question in community assembly, I found that, and mathematically I use something called the Akaike Information Criterion Score, AIC, but this is something even by eyeball you can kind of do, like you can look at how these things decay with sample order, is that you compare the exponential model with the power law model, and power law model just, it holds a lot better. So the more negative your AIC score is, by the way, relatively, the more likely the model. Power law model was much, much more likely, which meant that we were tending to see very strong evidence of niche differentiation with our invertebrates, which means that functionally, if this kind of invertebrate's here, it influences the likelihood of another one showing up. It is not just random whatever shows up. So these are the kind of results I found. Now, when I go back to all those zeta diversity parameters I calculated, now I wanted to make a model of the health of these streams at the watershed scale. So basically saying, like, are watersheds healthy or not? So I made this model. I used 6 parameters initially. So zeta 1, 2, and 10 for taxonomic diversity, and then another 3 for the functional diversity. And I built the model. So this is a simple linear model with these six parameters. And I said, okay, how correlated is this against the average California streams condition index value for your samples in that watershed? So basically, I'm just looking at zeta diversity patterns. I don't even care what the genera are. This is kind of the thing that was really interesting to me, is like, I don't even care what they are. I just want to look at the patterns. They could have been like piles of M&Ms. All right. So built the model, compared it against sort of this established metric, which is based on like specifically, do we see this? Do we see this? Do we see this? And this is what I found in terms of trying to correlate the two against each other. So on the bottom is the average health index score for those samples in a given watershed. On the y-axis, I have the index value that I calculated. R squared is like 0.85, which basically means if I just know the relational patterns and like how similar things are, how different they are, I can explain 85% of the variance. And that was a big takeaway for this first paper, which is really interesting to me. It's just like, I don't even know what they are. I just know, does this look like this? And that explains like 85% of it in this case. And they both follow, as you see, it's color coded by land use. So you just tend to see the things get healthier when you have less land use, which isn't too surprising. Both models behave very similar to changes in the environment, which was like another thing that I looked at. So anyway, just to summarize all this, is like we found that at least when we look at land use as the stressor, we get local diversity declines, but everything declines in its own unique way, the Anna-Katerina principle. We saw strong evidence of niche differentiation, which is if species A is here, it influences the likelihood of species B showing up by its role in the ecosystem. We show that we can use zeta diversity as a framework to potentially look at evaluating the health of ecosystems on this sort of regional scale. And we can also look at environmental changes that influence the presence and absence of rare species and common species, because we're looking at all these different orders of zeta. All right.
So colleagues, by this moment, in normal format of defense, we would probably have lots and lots of questions already. And we just let Levi speak for 40 minutes without interrupting him, which is very troubling. So because we are done with the paper 1, do you want to open it for questions for that paper number 1? And there was at least one comment in chat that Levi might want to address now.
Oh, where's the chat? Okay. Raphael at Squirp says they have more data. I know, Rafi, I'm actually looking at all the coastal sediment data too. I wonder, okay, super interesting. I wonder if the asymptotic fall off you found could be used to identify ideal ratios of land use to undeveloped nature. In other words, could we use this to make urban areas more biodiverse? Okay, I will get back to that by the end of this talk, because what I'm going to be doing next after grad school partly looks at this question. So thank you, Jen, for posting that question. I will address that. It's probably going to be what I'm working on for at least the next year after this. Okay. Okay, next question. Wondering if you took the data and put season into it, if you're able to find non-random associations. So for the seasonality question, Squirp does this they do a standardized practice where they get all their samples within a very narrow window during the year. So it's late spring, it has to be within a certain amount of time after the last major rainfall event. So everything, seasonality is like, they try to remove that as a factor. Nate, I'm not sure how zeta diversity, the R squared equals 0.85 lets you get away with obtaining taxonomic diversity by hand. So that's a good point. It doesn't let you get away with it, but what it shows is that we're just looking at the relational patterns by category. And when I'm trying, so the argument I made and sort of I actually finished off that paper with is that we could get diversity. In this case, it was done by hand, that taxonomic data. But I made the argument by saying, like, look, I'm just looking at relational patterns between categories. So plausibly, it doesn't matter if these are categories that were, built by hand or if they were built by doing metabarcoding and doing the sequencing and just putting things in piles based on that, because that will also just produce a series of categories. So that was the argument I actually started to make in that paper, because I wanted to write that paper in order to justify what we were going to be building to next. Non-convergence is interesting. I was trying to get to if the first settler influenced who was successful in different streams against the non-convergent question. So that is a really interesting question. For that, I would need to have longer-term time series data at a series of stream sites. So that's actually a question I would be interested in looking at down the line using long-term ecological research data, so the LTER data sets. And I think that's something that we could start to look at these kind of questions. But this was much more just like, a lot of this data was not repeated geographically. It was not repeated geographically at all. So I couldn't really address it. Uh-oh. I have some seasonal data, 5 years, 95 stations, we can talk. Okay. I like data.
Okay, let's keep moving on.
Okay. So back to networks, because I had that sitting in the back of my mind from 2003, and now I got to play around with networks. You're probably wondering, when am I going to talk about networks? Now we're going to talk about networks. So now, instead of looking at just overlap, like do these communities look similar or not, now I wanted to look at, well, if I see one genera show up, how tightly associated is its presence going to be with just another genera showing up in my community? And this is where we start getting this idea of ecological networks. So before I get into, there's all sorts of types of ecological networks. I'm going to be talking about what are known as co-occurrence networks. And I want to throw this preamble out because I know somebody's going to bring it up. And as co-occurrence networks are not food webs. They are not interaction networks. I know this. I've had to justify this many times to reviewers. I know. Basically, it's the same thing as saying your networks on Facebook are not the same thing as your real friends. And we all know that. But we use them a lot in ecology because they're actually a lot easier to generate than interaction networks. We're saying like what eats what, because to do those networks, you have to have people sitting out there for a long time and taking notes of like, well, did this thing eat this thing? Or did this thing pollinate this thing? Whereas with a co-occurrence network, we can get away with using much easier data, which is just, hey, did we happen to see these two things show up in the same place in the same time? So again, it's just, people use these networks, like generate these networks because the data is easier to collect. And for a lot of data sets, it may not even be plausible to ever really collect that specific interaction network data, especially if you're looking at microbial communities, you have thousands and thousands of them. You're probably going to always just be dealing with co-occurrence networks. I mean, in 20 years, we might have a way around that, but for the time being, this is probably what we're going to be doing. So it's only a maybe network. So we're just saying, like, do I happen to see species A and species B showing up in the same site at the same time? So there are a lot of different ways to make co-occurrence networks. I chose really actually the most conservative approach. So this is just by presence absence. Do I, like, not even how many, do I see this species, do I see this species? Or do if I see species A, do I not see species B? So I do either, are they likely to co-occur or are they likely to, one's gonna repel the other? So just like, and that's just by presence absence. And the approach, and this is by Moretta and Holm, what they did is you look at your observation data, so this is species by site, or it can be genera by stream sample or whatever, and you see like, where do you see all your species and at what locations do you tend to see them? And that's your actual observation data. And then you just say like, well, now let's just randomly generate just all these random communities using those same genera, but now we're just saying like, maybe there's no sort of pattern whatsoever. So this is our null model. So just like, maybe it's just random noise. And then what we do is we look at how likely, because you have a bunch of observations of all these species, of species A, B, C, and D, and so on. and you have, let's say, 20 or 30 sample sites that you look at. And so statistically, you can infer how likely you would tend to see A, then how likely are you going to see B, or if you see A, how likely are you going to see B excluded? And you compare your co-occurrence or patterns against for your actual observation against those generated from your random data. So again, now we're looking at co-occurrence for actual data versus observation. And we can use this to start to look at how likely your co-occurrence patterns are non-random. Like how much do they deviate from just noise? Because this is sort of the perennial question. Anything in science is like, how do I know this is just not just random junk? So, and using the standardized effect size score, we can look at how likely, how strongly this is deviating from just sort of this null distribution. So blue in this case, by the way, is positive co-occurrence, red is exclusion. How likely is this deviating from random? And then we filter out these standardized effect size scores by their significance. So you can set your significance threshold. In this case, they said like, okay, almost none of these were actually significant. And this is just between species A and species B, for example, or species 1 and species 2. And then you just build an edge. So what you do is you build networks where each node is a species or a genera or a family. And each edge is the likelihood they are going to co-occur or counter co-occur, or essentially or repel each other. So one's going to exclude the other. And you build, this is just a network. It's a very simplified diagram. And it's just based on like, do I see these things in the same place at the same time or do I not? And you build these networks, and you could do this. I mean, it would be like if you just stood out on a corner and you just watched people walking by every day, and you just make notes of people that walk in the same direction. You just say, oh, they co-occur. And if they go in the opposite direction, you say like they're counter-occurring, and you start making networks based on those associations. It doesn't mean that it actually means anything, but it may point to something that you want to investigate in the future. So this is the kind of approach I use to construct these networks. In this case, I'm using the genera that were in the streams, and I have my thousands of samples, and I wanted to see like, what do these networks look like, and what can I tell from them? So instead of just looking at just overlap between communities, now I'm like looking at just the edges between genera and the structure of those networks. So going back to that research group in 2003, and all these things the patterns that started looking at that show up over and over again, I was interested is like, are there some universal patterns that show up in these kind of networks? And can we use them to say anything ecologically? So people have been looking at these ideas and looking at various kinds of networks, co-occurrence networks among them, to look at ecological patterns, do analysis. There's all sorts of structural properties of networks that we can go into about this. But one of the reasons, again, is like we use them because for a lot of systems, it's like the easiest way we can get the data. So for example, here, like this is a simplified network, but if you have a co-occurrence network, for example, with just like these 9 species, and you see that they tend to cluster like this, it may be evidence that one cluster is one set of ecological relationships and another is another. And you have this one generalist species, maybe it's a pigeon, that connects both communities because it can make connections with kind of everything. It's a generalist, not a specialist. These are the kind of things that people can start to investigate with these networks. So the topological measures I looked at, and by the way, there's tons that I could have looked at, but the ones I want to look at, because these are the ones where I could find at least some decent background on ecological associations people have made in the past from these measures. And I wanted to also use measures that were not going to be as strongly influenced just by the size of the network. Because remember, like, if I have 1,000 species or in one that I use to make one network and 100 species in another, it's going to change the actual size of the network because I'm just going to have less nodes. So I want to choose measures that are not going to be skewed strongly because of the size of the network. And I want to make sure I'm looking at stuff, again, that's just not random. The measures I ended up using, so the mean association strength, which is essentially, this is like the edge weight here. This is how strongly, like if I see A and B, how strongly associated are they? So it's just essentially your correlation. So the higher that edge weight, the more strongly like the species A and species B tend to co-occur or at least competitively exclude. The connectance in a network is just a measure of just sort of the density of edges. So every edge here is a significant correlation, and there's a maximum number of potential edges you can have in the network. It's, by the way, if you have n nodes, it's n times n minus 1 over 2. where you can have a network that's completely saturated with edges. So everything's connected to everything. But you're probably not going to get a network that looks like that. So it's basically the number of edges you see divided by the number of edges you could have, and that's your connectance. The modularity, there's lots of different ways to calculate this, but how strongly is your network separating into subnetworks? Okay, so if you were to look at social networks, it would be how many clicks do you see in your social graph, essentially. Those would be your modules. And then there's this other measure, it's kind of weird, but it's called degree heterogeneity, which is how skewed is your distribution of edges in your network? So that is, do you just have a handful of nodes that have almost all the edges? Or is everything relatively evenly distributed? So these are like ways that we can start to talk about measuring networks. So the first one, and this is actually, by the way, this is using Genera. This is just a toy network, but this is from Genera in the kind of data I was collecting. And then I just wanted to show this is like with blue for co-occurrence, red for exclusion. And we use the standardized effect score in the way we calculate it as our measure of like how strongly are they co-occurring or not. So that's our one measure. This designated as E. By the way, it's E because it's edge weight. Another paper used that term, so sorry. Okay. Connectance, again, so how many edges you saw versus how many you could have. So this is an example. This is simplified network 5 nodes. This connectance is 0.5. This one's sparser, it's 0.2. Then we have higher one here, 0.7. And your maximum number of edges, by the way, if you have 5 nodes, is 10 possible edges. And this, by the way, is assuming, for those of you who are going to ask about with networks, there's no directionality in these networks. It's just simple co-occurrence. So I'm not saying like if A, then B. It's just A and B have this relationship. But I don't know what the directionality is at all. And I also don't deal with self-interaction terms because we're not dealing with interactions. So again, so modularity. This is an example of a network that has a high degree of modularity and things are pulled apart into 4 separate modules. They're of interest again in ecology because you will tend to see things partition into sort of these functional groups of things. These things tend to co-occur strongly with each other, but they only may be weakly interacting or co-occurring with other groups of organisms here. So it happens in social networks, it happens in ecological networks. Degree heterogeneity. This one was really interesting to me because this gets more to the kind of stuff they were doing over in Notre Dame, but it's a way of saying how skewed is your distribution of edges. And here's a way to look at it is, again, sorry, this is confusing, but the papers that talk about this also use zeta, because I guess there's not enough Greek letters. But if you have a random network, it looks something like this. It's very homogeneous. Like every node kind of has roughly the same number of edges. And your degree heterogeneity tends towards being some low value close to 0. But in networks where you have this structure where it tends to be heavily skewed, where a few nodes tend to have a lot of the edges leaning towards them, then zeta starts to grow, your degree heterogeneity starts to grow relatively quickly. And this is of interest for all sorts of reasons, but one of the things why people look at this idea of degree heterogeneity is that with networks, is that a random network is very resilient against random removal of edges, so random failure. Whereas if you have a very skewed network, it changes the whole dynamic where most edges are not very important. And if you happen to take out a few of the key nodes, you blow everything apart. By the way, ecologically, these kind of things are referred to as keystone species. It's the ones that if you remove, everything falls apart. And in the internet, the way to think about this is these hubs are things like Facebook and Google. Everything points to them. If you remove them, you completely splinter the network. But these kind of networks are actually very resilient against random attack because most of the edges are not important. By the way, the internet was set up this way because it was deemed to be the most resilient structure against nuclear attack. So I guess you can't nuke the internet. All right. So I ended up looking at all these measures here, and I generated a series of hypotheses of what I thought the relationship between environmental stress, which by the way is going to be land use again, and these measures of our networks. Again, this is at the watershed scale where I'm taking like all these communities and saying, what do these ecological networks look like? What are these topological measures going to do? Can I make any predictions on that? I made the following 4 hypotheses. So the first one is just this idea of connectance. And this is, hold on a second here. So my first thought was connectance was going to be positively correlated with stress. And the reason I thought this is because under stress, you tend to get more generalist species. Your generalists are things like cockroaches, things that can survive anywhere under almost any condition. Generalists will tend to co-occur with other generalists because you tend to find them everywhere. If you were to do a co-occurrence network with pigeons, pigeons would show up next to everything. They're kind of like humans. They'll co-occur everywhere. So I thought that under stress, you would tend to only get the generalists left, and so the connectance would go up. Even though the number of nodes may be lower, the connectance is going to go up because all you have left are the generalists, your cockroaches. Okay. The co-occurrence strength, what I thought was going to go with that is I thought that was actually going to get stronger, which sounds weird because you think like, wouldn't stress cause ecological relationships to break down? The reason I thought is that the only things left though are going to be generalists co-occurring with other generalists. So even though lots of the edges have been removed, a lot of the co-occurrences have been removed now in your network from these communities under stress, the only thing left are going to be your cockroaches, and cockroaches will tend to co-occur with other cockroaches. So you'll have these, you should end up with really dense networks with a lot of strong connections, but between the kind of things that can survive the apocalypse. For modularity, I thought that my hypothesis was that it should be stronger. So you should have the stronger partitioning of your network, your co-occurrence network, into smaller subnetworks, because you tend to see greater dissimilarity between communities. So think back to that zeta diversity paper. Everything kind of became more different. If it becomes more different, that means that everything's going to should separate out where, oh, these things are going to co-occur here, but they're going to look a lot different from these things over here. And so whatever connections are between them are going to be a few and far between, things that can kind of hop back and forth ecologically between these like strongly partitioned and now like relatively disparate communities. And the last relationship, this is kind of weird, but it's the argument I made, is that the degree heterogeneity should actually go down with stress. So that is, you should have a more even distribution of edges between nodes. So your network should look more random. And the reason I thought that is that if connectance goes up, if your network looks more saturated, where like, if your connectance goes up, it's a smaller network, but everything that's left tends to co-occur with everything else. But if everything tends to co-occur with everything else, then your edge, all your edges kind of look sort of evenly distributed across your nodes. It just turns out to be a very saturated network, but a very uniform network. So these are hypotheses I made. Now, how did I test all this? Well, this is a lot of work. What I did is I took watersheds that had at least 30 stream samples. I grouped everything into land use quintiles. So like looking at just different, essentially making different stress categories. And then what I did is I would randomly then take 25 samples from each of these watersheds that happen to be within a particular region, or in this case, a particular watershed with a particular level of land use. So it could be the Los Angeles watershed, and those that happen to have a land use, you know, let's say like 25% to 100% coverage. So that's going to be like your heavily degraded areas. And so I made all these groupings, and I did the same random permutation where I would subsample and then do this and generate a network with each group of 25 samples. So like picture, I have 25 sites. I have all these hundreds of species, or in this case, genera, sorry. And then I would generate the co-occurrence network each one of those times. And I did this, and I ended up generating through this permutation process close to 4,000 networks. And this took a long time, by the way. Thankfully, USC is very generous with their computational resources. So I was able to do this. It only took like a week and a half with 120 gig of RAM. So this is, by the way, this is the same kind of data I used to do the first data diversity paper. And this is what I got for the samples I was able to use. And what did I do with this? So correlate everything at once. And this involved breaking up all of my data in like 500 different processes, writing all the 500 ones in parallel. And then, a week and a half later, everything came out. And what did I find out? I was actually really shocked by this because I really felt like I was flying blind in a lot of ways. You know, I found papers and I was kind of trying to tell myself like, okay, I should see this, I should see this. But it was like one of these things where like, this is so weird that I kind of didn't expect a lot of things to be related. And it turns out that my hypotheses are all supported, which was kind of frightening to me. So the p-value, by the way, is 10 to the minus 4. Everything's less than this, so that means that there's a 0.1% chance I'm seeing this just by randomness. And the correlations all bear out. Now, they're not necessarily all the strongest correlations, but I did find at least some evidence to support all 4 hypotheses. And to give you an idea of like, what do these networks actually look like? And I removed, by the way, the names because it would just become too crowded, but each node here is a genus. And the edges here, they have different thicknesses depending on how strongly they're co-occurring or not. Blue is for positive, red is for exclusion. And so then the question is like, okay, well, which one is the stress network and which is not? So given what I all said, good question, me. All right, this is what we found out. So this is, by the way, like I ended up just graphing some of these networks to see what they even look like. And stress networks, this is actually a really typical example. This is what a stress network looks like. You have these tightly interconnected clusters that only have a few connections between them. So they have this high modularity. They tend to have really high edge weights between them. And every node tends to be pretty well saturated with connections to every other node in these like small sub-connected modules. Whereas in a low stress watershed, you have a huge distribution of edges. You have lots of weak co-occurrences. You have a lot of strong ones. You have a whole distribution of edge weights, and you have like a lot of different connection structures here. You also have a greater degree of, you also have a higher degree of degree heterogeneity because you have a few really connected nodes here, but a lot of things that only have a few edges. And so this is the kind of thing that I saw. Now mathematically, like I bore out just what I showed you right before. This diagram again. So this is what I was thinking like, well, okay, what might be the mechanism? So this is where I thought like, again, it may literally this may be a reflection of the same thing we saw with the zeta diversity patterns, again, with the same data set, where everything pulls back in terms of its taxonomic overlap or functional overlap. Everything has lower local diversity and everything looks more different. And these co-occurrence networks may just be a different way of reflecting the same exact ecological pattern. In physics, we like to say this is conformal, which anyone who's ever dealt with conformal mapping, good luck. All right. But this is what I thought like may be going on here. This is my best guess. So local richness declines. There's actually less convergence in community composition. And I think this reflects not only now with Hawkins, but now I get to cite myself, which is always a great moment in anybody's life. It's like, I think this also reflects what I saw with zeta diversity. So that was my best guess. This is what I think might be going on with the networks. And this is like, there's some other things I looked at with this. If we could use topological measures to construct indices of ecological health, the answer is similar to zeta diversity, you can. But I think I was just very interested that we saw any sort of significant connection between these topological measures and the health of watersheds. And the reason I'm surprised is think about how many levels of abstraction we are at this point. So we're starting with all these just invertebrates across all these different streams and all these different watersheds. And all I'm saying now is, hey, let's make networks that show, hey, how likely if I see A, do I see B? And I'm not even looking now at these networks. Now I'm just looking at these measures of the structure of their topology. And just that alone tells me something about it. It would be the equivalent of if I just took social graph data from Facebook, removed everybody's names, looked at the structure of those networks, and I can tell you what the GDP happened to be for that city. That's what it felt like to me. So maybe it was a fluke, but this was something that was really interesting to me. All right, so what do we learn? Okay, we have smaller networks under stress, they're denser, they tend to be stronger patterns of co-occurrence. My guess is generalists tending to co-generalists. And similar to zeta diversity, this may be pointing to a way of saying like on this regional scale, we can look at these patterns and say this system is degraded or it isn't. All right. Oh, wait. All right. So I'm going to go on to chapter 3, but before I do that, there's something in the chat. Okay.
Yeah, and apologies. At this moment, I would like to ask committee members what colleagues would be your preference, because Levi is already speaking for an hour, and normally this is a limitation of presentation, but also it is Zoom presentation, so it is a little bit it is a little bit slower, as you know from teaching classes over Zoom. So what would be your guidance?
Well, I'm here for a while still, so I'd be happy to hear it, but obviously don't want it to go much longer because we have the audience, where the public is supposed to have access to this as well throughout before we get to our questions. So how much longer do you plan to go, Levi?
Probably another 20 minutes or so.
That's pushing it, but I guess it's okay.
Oh, all right. I'll try to be quick here. Okay.
As a committee members, any concerns, or shall we just go on?
Yeah, no.
I'm fine if Levi just wants to wrap up in 15 or so.
Okay.
Yeah.
All right. I'll take that as a challenge.
Okay.
Okay. So now we actually have our metabarcoding data. So I did this with all the archive data. Now I want to test out, and now I looked at zeta diversity patterns with the metabarcoding. So now I want to say, I now want to look at not just invertebrates. Now I want to look at diatoms, algae, bacteria, and invertebrates. And I want to see, like, can we pull this off using metabarcoding instead of stuff that's sort of taxonomically, to come back to Nate's question. So I showed that diagram earlier. How does this middle part work though? So this is sort of like step 1, step 2, question mark, step 3, profit. And this is one of these really great techniques now that we have in molecular biology. There's certain these snippets that you can take from genomes of organisms. These are called these barcode sequences. So like 16S, 18S, there's lots of different types. But we can use them to very rapidly classify with just a few hundred base pairs what happens to be there in the environment. And so this is what we use. We used a number of these different what are called primers to look at different communities in our streams. So this is a much smaller set of samples, but it was a much deeper set of samples because now instead of a few hundred genera, we have thousands and thousands of species of everything from bacteria all the way up to invertebrates. So 165 samples. This is 2 years of data. We looked at eukaryotes, so things like you and I, using 2 different primers. So this is an 18SV9. And then we used an RBCL primer to look at just diatoms specifically. For prokaryotes, we used 16SV4. We used one that spans roughly the V4 to V5, using a primer actually developed by the Furman lab. And we use the same measure of stress. This is, again, 5 kilometers upstream. And these are where our samples are located in the measure of land use. All right. Now, how did I process this time? So I have 3 different land use bands. So pristine is anything that's like 0 to 3% coverage, 3 to 15% is intermediate disturbance, and anything above that, we're going to call degraded. I calculated zeta 1, 2, and 10 for all these taxonomic groupings, and I did this for different levels of taxonomic aggregation. So I said grouping like the communities at the level of species, genus, and so on, because I wanted to see how do these relations, do they hold depending on how much we like, how finally we resolve what our communities look like. And I did the same thing as the first paper where I said community assembly models. And this is done, I did 100 random permutations of this. So I ended up with 300 sample groupings. So not nearly as big as like the first one, but it's way, way more depth in terms of our community data. And long story short, what did we get? Let me explain what this is. This is comparing the AIC scores for exponential versus power law. So power law scores are going to be in parentheses. The more negative, though, of the two is going to be the more likely of your model. And what we found was, for the most part, except for a few here with these benthic macroinvertebrates and for prokaryotes using this primer, for the most part, we saw that we have way more negative AIC scores for the power law model, which means we have much stronger evidence of niche differentiation for all of our communities, independent of like what level of taxonomy we're zooming in on, for the most part. I had some ideas of like why that might be. Maybe we're looking at something that BMIs or benthic macroinvertebrates are randomly distributed level of species, but everything gets more sorted out via niche differentiated level of family. But my suspicion is it's a limitation on how well we can assign taxonomy given our snippets of DNA, because depending on how well we can resolve those snippets, we may not be able to specify what we're looking at any better than the level of family or genus. So there's a lot of snippets of DNA where I can tell you what family it's in, but I can't tell you what species it is. And so we may be losing a lot of resolution, and that's changing what we're seeing with some of these communities. This is a kind of quick data table that shows you that. But for the most part, so ASV is the amplicon sequence variant. This is the number of unique snippets. And we see really sharp drop off here for, let's say, with bacteria, where we just don't know them very well to the level of species or even a genus. Some of them we know very well. So benthic macroinvertebrates, we had a pretty good handle on, and diatoms, but we know diatoms only really well with this one particular primer. So everything's kind of really evenly distributed. And went and constructed this index again. Now I use Zeta 1, 2, and 10. This time just with the taxonomic data, that's what I had. And again, using 25 samples per land use band and calculate against the average stream condition index using all these different communities. So not just benthic macroinvertebrates in this case. And this is how strongly, this is the correlation I found. So the Pearson correlation, this is between my indices I constructed for my communities. And then within the parentheses, what I did is now I did a 10-fold cross-validation where I just randomly split everything into 10 subsets of data and then see how consistently do these correlations hold. So you're gonna lose some of your predictive power. And what I found was actually kind of surprising is that benthic macroinvertebrates do perform all right, but we found much better results in our system using diatoms. And I wanted to see how much of an influence these different orders of zeta diversity had in these models. So I wanted to look at their relative importance. So remember, our model is Zeta 1, 2, and 10, but maybe almost all the change in the model that we have is due to just local diversity, so Zeta 1, or maybe almost all of it's due to like Zeta 2 or Zeta 10, which is just relational patterns or the overlap between these communities. And we found significant differences in how this was structured with our communities. We found with benthic macroinvertebrates that, let's say, we saw a lot of variation here. It skews more towards local diversity in a lot of cases. It may change as we go higher up in this, like more coarser taxonomic groupings. What was interesting to me, though, diatoms perform really well, but no matter what order, no matter what level of aggregation we looked at, we saw a really even distribution in the relative importance of these different orders of zeta in our models, which means that we're seeing something that with diatoms maybe, that they're capturing changes not only in the local diversity, but also to a very equal degree, they're capturing something similar with Zeta 2 and Zeta 10, which is just landscape diversity as well. And they're doing it just across every level of taxonomic aggregation. And it's interesting to me because the benthic macroinvertebrates are initially, that's what we were used to calculate the California Streams Condition Index in the first place. And yet, at least using metabarcoding data, diatoms actually are better at explaining this index than even the benthic macroinvertebrates. And so why that may be, or if it's just a fluke, is something that I'm really interested in addressing. But that is actually what we found so far. What we did find, though, is that, again, even if we use these metabarcoding sets to construct our communities, as opposed to doing it by sorting by taxonomy by hand, is that we found that we can still use zeta diversity as a pretty good predictive tool of the health of streams on a regional scale. One of the things I also found is that there's a lot of limitations with trying to assign taxonomy to sequences. This is not a problem that's unique to me. This is something that comes up in a lot of research involving environmental DNA, especially because there's a lot of things, bacteria and archaea especially, where we don't really know what the sequences are supposed to look like. And so there's a lot of work now being done on what are known as reference-free techniques, where we can actually look at just patterns in group sequences, like snippets of DNA, without even knowing what they are, and just saying like, these snippets are probably all in the same species. I don't know what it is. And if you're just interested in looking at these relational patterns, that's actually good enough. So for future approaches, that's a technique. And so this kind of leads me like 3 years on, okay, landscape ecology, but why do I see all these patterns? Like, stuff seems to work, but why? And it's something that I want to keep pursuing as a career after this. I don't know, like I said, maybe it'll turn out this was all a fluke. I'm hoping there's probably some meaning in this. But again, coming back to my bias is like my background in physics and thinking that like, okay, everything's kind of governed by the Taco Bell menu of cosmology. Maybe this is the lowest energy state. And well, this kind of want to leave this off on the most inspirational quote I can think of. But maybe the universe is just approaching this as the lowest energy state. This is how things are going to show up in terms of diversity patterns. because this is the easiest way for it to work. Of course, question of that is, why is this the easiest way to work? But it's something that I really wanted to investigate because, again, it's like my bias, but at the end of this, at the bottom of everything, you still have to deal with thermodynamics. And so why things are the way they are is probably because it's the easiest way for them to be. But why that happens to be the easiest way to be, that's probably gonna be the rest of my life. So before I finish up, I do wanna mention other people I collaborate with, and it was actually a lot of fun to do this, with stuff that has nothing to do with my thesis. But these are papers that I was able to produce. So first I want to acknowledge Nate here. This is why I was going back and forth to the island a lot back in 2017. But we did a lot of work with oysters and aquaculture, and I got to learn a lot about the oyster gut microbiome. So we got this paper out back in 2018. I did a lot of work with Travis Longcourt here on looking at landscape ecology and light pollution. And so I think some of the students who worked with me on that are on right now, where we got to drive up and down the coast at night, taking thousands of photos of the night sky with a fisheye lens, and that was a lot of fun. And we got this paper out actually this year. We're working on a second one, applying this to looking at species distribution. And we have this third paper. This is Mei Shi over at UCLA. This is actually using eDNA, and I did zeta diversity analysis with the eDNA with the Cali DNA group at UCLA. This is in review right now. I have reviewer comments sitting in my inbox right now. I will totally get to it, I promise. But that is tentatively the title of that paper. So I've had a pretty good run so far. Anyway, and why am I doing this? Okay, so long story short, I'm gonna be going to UCLA in the fall. I'm gonna be doing a postdoc for at least a year with the Institute of Environment and Sustainability at UCLA. And I'm gonna be looking at this site called Sage Hill, which is totally undeveloped land in West LA, which is really interesting as a test case for these ideas of ecology, especially in an urban context. I'm gonna be teaching in environmental science there. And I'm also gonna be working on, to answer an earlier question, looking at developing methods for assessing the health of ecosystems in Los Angeles. So developing something like the California Streams Condition Index, but for landscape health in the LA area. So this is of interest to the Los Angeles Department of Environment and Sustainability. So I'm gonna be working on that. And I have a couple of teams of seniors who are gonna be working on it as well. So that is where I'm at. So hopefully I was quick enough here. So thank you. Thank you for coming out this afternoon or evening, depending on what time zone you're in at the moment. But okay.
Thank you very much. So let me open it to questions, first of all audience, and then we will move to committee members. Questions, comments, concerns?
Oh, are people going to send it on chat? Oops. Ah. Okay. No one wants to say anything?
I'll speak my question.
Okay, thank you.
So in regards to chapter 2, the network modeling, would it be possible to use those findings to simulate what might happen in stressed and non-stressed networks? Let's say you delete certain nodes or connections.
Yes, I think that would be. I think it would be more feasible to do that with data where we have repeated observations of the same sites. So that's actually been one of the limitations of the kind of data I've looked at is it's very geographically wide, but a lot of times like they're not ever repeated. I have an interest in looking at sediment core data because that'll give me some opportunity to look at trying to do ecological reconstruction at a location over deep time and start to look at these network patterns over really long amounts of time. But for the kind of data I have, I think it would be, I don't know how feasible it would be. But yeah, that's a really good question.
Great, thank you.
Thanks, Jen.
Any more questions? I guess, and it means that we should, committee members and Levi should go into a breakout room and we could ask Levi some questions and grill him a little bit longer.
Okay.
Except I have no clue how to establish breakout room. Anupam, do you know how to do it?
I can figure it out.